This is an ordinary corn seed. Corn is produced by a monocotyledonous plant, with each seed normally having one embryo containing a potential root and shoot. If allowed to germinate, it normally produces one shoot or a single seedling. However, in some cases, corn embryos have germinated to produce two or more plumules or shoots, each having their own primary root system from the single embryo. Since they are not from two or more independent embryos, but from a single embryo, this condition in corn is often called false polyembryony. True polyembryony is defined as the development of multiple embryos within a single ovary, seed, or fertilized egg. In plants, it plays an important role in genetic replication and diversity. Polyembryony in maize or corn have been reported and described as a complex, elusive, and a potential agronomic useful trait. Hello everyone, this is Raymond from Best Fruits in Jamaica. Recently, for the first time in my life, I accidentally came across a single germinated corn seed that produced two polyembryonic corn seedlings. And for those of you who might not have seen such a wonder before, I feel obligated to share the first set of photographs that has ever been recorded at this stage of this elusive natural phenomenon with you. I like to grow sweet corn in my home garden. However, the soil around my home is moderately infested with mole crickets or cutters, and I have found that sweet corn seedlings are among their favorite meals. I try to avoid using any insecticides on very young corn plants as they sometimes display toxic reactions to some insecticides. In addition, there are a number of beneficial insects or predator insects in my area. Not using synthetic chemicals within the area will naturally preserve the population of these beneficial insects, as they are sometimes the greatest casualty whenever insecticides are used in any area that they occupy. I usually start my corn plants in pots to protect them from these mole crickets or cutters, and when they are about 2 weeks old, I transplant them into the field. For this set, I had pre-germinated them in a saucer with some water to start the germination process instead of planting straight into the pots. In just over 12 hours, there were signs that the root system had started to emerge. So I planted them out in the pots. Germination of a corn seed involves three stages. One, imbibition of the seed by absorbing water. Two, respiration or the breakdown of stored food in the seed. And three, cell division and differentiation or reorganization of cells into the root and shoot system of the plant. Germination rates are influenced by available water and soil temperature, as days to germinate can range from 18 to 21 days at soil temperatures of 50 to 55 degrees Fahrenheit or 10 to 13 degrees Celsius, or 8 to 10 days at temperatures over 65 degrees Fahrenheit or 18 degrees Celsius. However, in hot tropical conditions, it is common to find corn seeds germinating in 3 to 5 days in temperatures over 90 degrees Fahrenheit or 32 degrees Celsius. While planting them out in the pot, I did notice that the germinating seed that produced the two seedlings was different at first, but was not sure of the reason. However, I did place it alone in a small pot, after which all were placed in a semi-shaded area, which provided some shade during the hot days.
Since I usually plant about 50 seeds each time,
I usually water them every morning when there was no rain the day before, or the rain was insufficient, and if it was adequate, I would still find the time to observe them. And each time when I quickly observe each growing plant, the single seed in the pot would get extra attention. On observation, on the fourth day after they were planted out in the pots, I noticed that another seedling had appeared in the pot that I had planted a single seed. Further observation 2 days after confirmed my suspicion that both seedlings were slow growing when compared to the other seedlings, even though all seedlings were experiencing the same growing conditions. At this time, I was still not certain that they were polyembryonic or two different seedlings from two separate seeds. The growth rate of these particular two seedlings were extremely slow, so I decided to investigate further by removing them from the pot. This I did by removing them with the soil. Then they were gently placed in about half an inch of water in a small container, which slowly caused the soil particles to separate from the roots, revealing their polyembryonic characteristics, at which detailed observations were made. Both shoots were separate, with each individual seedling having its own root system. Because there were two seedlings feeding from the same endosperm, it was normal for those two to be smaller than the other seedlings in the group. Since the stored food had to be shared for two plants in this case, but only one for the other seedlings, it was clear that there was competition among both seedlings as one was shorter and less robust in growth than the other. I was able to obtain a bit of magnification so that some of the details might be clearer, as you might not have been able to see them from the images made by only the normal lens of the camera. Here you can see the features of both seedlings, where they were attached to the corn embryo with camera alone and with magnification. Their shoots, individual root attachment with root ears, and in this case, how both were attached to the same seed. Corn has a fibrous root system comprised of a number of smaller lateral roots that spread out in all directions from the stem. After detailed observations and recordings, the seedlings were replanted in the pot for further observation. I will try to keep these plants alive. However, I do not have any special facility for doing so, but I do think that I could be back with a follow-up video, which will depend on the interest generated by you, the viewers, on the outcome of these two seedlings. I did learn from this experience, and I hope you did too, as it was the first time I had ever seen and held polyembryonic corn seedlings with my fingers. Thank you for watching, and please remember to subscribe to the channel if you have not done so before, and to like and share this video with your friends, families, and any students that you might know with a keen interest in biological related careers.